Got a full house today. That's right. Good morning, everyone. It is our distinct honor and privilege to welcome you to Grand Rounds at Massachusetts General Hospital. My name is Dr. Jonathan Crow. I'm one of the chief residents in neurology here at MGH, and we are so excited about Grand Rounds today. We are so excited to hear from Dr. Jonathan Jackson and to hear what it is that he's going to be sharing with us today in his talk. Dr. Jackson is the executive director of the Research Center at Massachusetts General Hospital and is an assistant professor in neurology at Harvard Medical School. Dr. Jackson is the executive director of community access, recruitment, and engagement, the CARE Research Center at Massachusetts General Hospital, and is also an assistant professor of neurology at HMS. CARE investigates the impact of diversity and inclusion on the quality of human subject research and leverages deep community entrenchment to build trust and overcome barriers to clinical trial participation. Jonathan's research focuses on inequities in clinical settings that affect underserved populations, and he has received generous funding for his work from the Michael J. Fox Foundation for Parkinson's Research, from Massachusetts General Hospital, and from the National Institutes of Health, including a prestigious NIH Pioneer Award to advance this work. Dr. Jackson, who received his PhD in psychological and brain sciences in 2014, also conducts research as a cognitive neuroscience in the early detection of Alzheimer's disease, particularly in the absence of overt memory problems. He has become a well-known representative to underserved communities in dozens of affiliated organizations, especially regarding the participation in clinical research. Dr. Jackson serves in an advisory capacity for several organizations focused on equity in clinical research and has written guidance for local, statewide, national, and federal organizations on research access, engagement, and recruitment. We are so excited and honored to hear from Dr. Jackson today as he gives us his talk, Quantifying Diversity and Inclusion in Clinical Research. Dr. Jackson, welcome. Thank you for taking time to be with us this morning.
All right, thank you so much. I will remove my mask so it's a little bit easier to see and hear me. Thank you so much for inviting me to be part of the Grand Rounds presentation. I'm going to be talking today about how we do this work about quantifying diversity and inclusion in clinical research. I think we are all on the same page in recognizing that it's really important, but we are not quite all on the same page in recognizing how we do this and how we measure any sort of indicators of success. As we get started, I wanted to kind of quickly step through my disclosures. This is a bit fuller of a disclosure statement than you normally see, but because we are talking about things that are really on the fringes of the way that we currently understand science, my hope is that you kind of know where the money's coming from. I wanted to start with a really simple and clear message, which is that historically, when we talk about diversity, equity, and inclusion in research, we come at it from a perspective of it being a moral imperative. This is the right thing to do from a societal standpoint. What I want to make very clear is that if we don't have representative research populations in our work, then it also destabilizes and undermines the causal inference that we use to draw conclusions from our research studies. So we can recognize that there are threats to the four major types of validity that we tend to recognize in research. And so in addition to there being major threats when it comes to external validity, we also know that there are limitations to construct, internal, and statistical conclusion validity without a representative sample. So if we don't recruit representatively, I wanted to show you a real world example of how that makes it a little bit more difficult to understand the conclusions that we're trying to make about a scientific enterprise. So what we've got here are a breakdown of the populations represented in the two main trials that formed the aducanumab trials in Alzheimer's disease, also now known as ADUC. So what you can see here, first of all, is that even though this trial covered around 850 participants in 20 countries. It was still overwhelmingly white. But what I want to do is zoom in on this particular demographic group. So these are Black and African American individuals, which is about 0.5% of the participants in the research study. So not enough to do any kind of comparative analysis or subgroup investigation, but it's really important to understand why we need to be a little bit more thoughtful about who's represented in our research studies. And in order to understand sort of the significance, the relevance of our lack of Black participants in the ADUHELM trials, we need to look at one other sort of major risk factor for Alzheimer's disease, which is what was studied. And for that, we're going to go back and take a look at the prevalence of APOE. So APOE is the major genetic risk factor for those of you who aren't steeped in the world of Alzheimer's disease. And typically, large clinical trials these days have between 50 and 90% of the representatives have some sort of APOE allele. So we know that one of the major side effects in current trials, we heard about that earlier this week with the lecanemab results, is some presentation of a sort of amyloid-related imaging abnormality, some kind of microhemorrhage or macrohemorrhage, or sometimes in rare cases, an edema. And so we can see that individuals with this E4 mutation tend to have a higher prevalence of these ARIA. So again, this is the aducanumab trials. We haven't seen the data for lecanemab. My guess is it's going to present somewhat similarly to this. Now, what does this have to do with race? The assumption is that we know a lot about APOE risk of Alzheimer's disease, but unfortunately, we know the most about that risk in white individuals. If we break down who actually has that E4 mutation, we recognize that it's actually Black individuals, followed by Latinx individuals, and then, finally, white individuals that tend to have this E4 presentation that confers a significant Alzheimer's disease risk. So what we are seeing here is that even though most individuals in Alzheimer's disease studies have this E4 mutation, overwhelmingly they tend to be selected from a population that has a lower prevalence of the E4 mutation relative to other groups. And so that is one of many factors that made it really, really difficult to interpret the data that we saw with the aducanumab trials. Hopefully, fingers crossed, with the larger diverse population that we saw in the lecanemab trial, we will be able to conduct some sort of subgroup analysis. But ultimately, what I want to highlight that is to sort of say, when we talk about diversity, there is a way to make a statistical argument. And more importantly, there's a way to connect that to the way that we currently think about scientific design and analysis. So historically, when we think about differences between groups, we used to sort of say that they must be biologic in nature. So Thomas Jefferson himself said, "Never believe a Negro." We know that the work of Samuel Cartwright in the 19th century showed that Black individuals, had smaller brains, and therefore this was an inherent trait. We all know, as people steeped in the world of neurology, the early indications of this work by looking at the different sort of kind of skull formations in our brain and sort of making essentialized differences there. So hopefully we have all moved past that and moved past the history of phrenology. And now when we think about differences between groups, we recognize that there may be some inherent difference, usually expressed in an epigenomic fashion, complemented by social, structural, and societal forces that may reinforce some of these underlying differences. It is my belief that this second presentation is still limited, and it may be better to think about things in this way, where we have a combination of those biological disparities and social inequities, but in addition, we have to be thoughtful about selection bias, measurement error, and implementation and design bias to not only for the sake of helping us understand diversity, equity, and inclusion, but for the sake of good science. So the point that I'm trying to make here is that by paying attention to aspects of representation, diversity, equity, and inclusion, you're not necessarily trying to fulfill a societal good, you are just trying to conduct rigorous, responsible science. And that is the take-home message of today's presentation. So we can actually look at the way that we can represent and understand limitations of diversity, equity, and inclusion as a function of all of these factors. So there may be elements of biased treatment or biased access. There may be some sort of epigenomic variance, but there is a lot that we tend to be missing. we tend to find ourselves failing to pay attention to these first three sources of variance. And ultimately, what we end up doing is rolling all of that variance into our error terms for our statistical models, which makes it harder to look for the thing that we're trying to look for in the first place. So in terms of how to solve some of these issues, what's really crucial and what's really critical here is understanding that there is no tension between robust statistic and analytic design and a truly representative, diversified research population. So if you think about the science of recruitment, engagement, and retention, that is a solution to a selection bias. We can also think about tools that are more familiar to some of us, such as differential item functioning as a solution to some aspects of measurement error. And then, as you kind of move down this framework, you can see that when it comes time to trying to resolve biological disparities or social or societal inequities, you can think about the way that we traditionally embrace community-based participatory research in our research designs. However, what I want to emphasize in today's talk is that there's a lot that we can do before we go out and try to convince community members to take part in our research that may enable us to conduct sound, rigorous science. And so today we're going to walk through an example from some early data and some things that I've done over the last year or so that really really drives home this concept of thinking about selection, measurement, and implementation in particular as a means to resolve challenges that we have in diversity, equity, and inclusion when it comes to research representativeness in our clinical trials. So I want to walk you through one seemingly controversial example and show you how the framework that I just presented, this equity as variance framework, helps you come to a relatively clear conclusion. Now, one question that has been raging over the last several years is, what do we do with race? How do we incorporate that into our research designs? How do we measure it? How do we compare groups together? So we have to think about how we typically use race in our studies. So we tend to use it potentially as a risk factor for disease. So in my home area of Alzheimer's disease, we will say that Black and African Americans, actually more African Americans than Blacks, are 2 to 4 times more likely to develop Alzheimer's disease relative to white individuals. Latinx individuals are 1.5 times. And this is usually attributed to comorbid factors of vasculature, though there's not necessarily strong evidence to back that up. We also use race in these ways when we're doing our research designs. We'll use it for dimension reduction. We'll use it as some sort of proxy for ancestry or genetic variation, or we'll use it as a marker for societal factors such as racism. Unfortunately, according to this equity variance model, none of these are appropriate or valid conditions for using it. So there's no justifiable basis for race creeping into our research models the way that we are currently using it for any of these 3 reasons. So, if we think about it this way, we break down understanding using race as a construct for dimension reduction. We know that it is a core and uncorrelated or undercorrelated proxy of socioeconomic status, and that's just because race doesn't have any strong empirical definition. If you're looking for like a sort of an easy example, just think about how race has been defined in each of the last 4 US censuses. It has not been consistent. So in 1990, the US Census had a set number of races. In the 2000 census, you were allowed to pick more than 1 race for the first time. In 2010, you were allowed to select multiple races. In 2020, we know that there was a massive undercount due to some fumblings by the federal government that was in charge at the time. So what that means is that our empirical measurement of race, usually based on US Census definitions and data, is not going to be strongly correlated with any sort of dimension that we're trying to reduce there. If we're going to try to use race as an empiric variable, it is not a really great marker of genetic variation in ancestry. It is a well-known fact that individuals tend to have more variation within socially identified races than between them. And so then we also need to recognize that if we use race as a variable, there tends to be this inadvertent baselining and norming of usually white identifying individuals. And so that sacrifices validity because there's no inherent reason to baseline your data on white people. And then obviously as an indicator of racism, it tends to erase bias and it tends to make it tends to reduce individuals to single sort of sociological or individual identity markers. It sort of shifts the focus on individuals from the barriers. So you're focusing on race instead of racism, for example. And it does tend to create misleading monoliths, such as all Black people experience racism equally. Which again does not provide you the variance that you need in order to explain some of the relationships that you're seeing in your models. So you might be thinking, okay, that sort of makes sense, but it's also confusing because in this new era of post-George Floyd, I know that I need to really pay attention to race instead of ignoring it. So let me break this down for you just a little bit more carefully. So what you need to think about is, let's be scientists about this. Think about your mechanism of action. In the vast majority of cases, unless you're doing some sort of facial detection or pulse oximeter reading, it is not going to be the amount of melanin in your skin. That is not going to be your mechanism of action. So do your best to measure what you think that mechanism is, whether it is a mechanism, environment, or exposure. Measure that rather than some poor proxy of it. Leverage more useful group differentiators. It's the year of our Lord 2022. We have gotten a lot better at trying to identify things like access to care, literacy around health, decisional authority. But make sure that when you're building your research models, when you're building your statistical analyses, be careful about your reference groups. Do you really want to normalize white individuals, or do you just want to normalize lowest risk or lowest relative ratio, for example? Now, what I would recommend is, instead of using something like race, to maybe consider a standardized set of social determinants. It gives you a much better opportunity to identify useful amounts of variance in your models. It allows you to see the hidden diversity, such as white individuals living in rural areas that tend to face a lot of the same sort of socioeconomic challenges that we usually sort of poorly ascribe to ethno-racial minorities. It centers exposures in systems rather than essentializing it to individuals. It does make recruitment much easier, and I'll get to that in just a minute. But what I want to emphasize is something that I found in my 10 years in Boston, which is race tends to be considered something of a third rail. But the good news is that we're scientists, so we can treat it like a scientific variable. Use it where it's useful, which is usually in helping us understand the demographic identity of our research sample. And then we need to sort of drop it from the rest of our scientific studies. So racial identity can really only ever be a marker of racial identity. If you're using it for anything else, you tend to be leaving variance on the table. So, lest you think I'm immune, there was a study done by this guy, Jackson, published about 5 years ago in Age and Aging, where we found that there was a difference between African Americans and Caucasians, yes, I regret those terms too, in terms of subjective versus objective correlations of memory performance. We found the expected negative correlation between the increase in subjective cognitive concerns with memory performance in white individuals, but when we switched to a Black population, we found that there was no strong correlation. When we took a sort of a deeper dive into the data, you can see that there were strong correlations within the variables that made up these constructs for whites, but not so much for Black individuals. However, what we ended up doing was we tried to match the sample and tried to see if we got a white population that was as similar to the Black population as possible. We were able to match on age, education, MMSE scores, and reading ability, but we then created sort of emergent differences, like in the geriatric depression scale. And ultimately, what that means is that we ended up committing a number of these errors that according to this model. So we ended up making mistakes with respect to selection bias. We mismeasured the construct in question. We ignored any of the variance that may have arrived from social inequities or biological disparities. The only thing we got right was we sort of minimized the implementation bias. But ultimately, although we did report this finding, and that group has since been able to replicate that finding, because there are so many conflations of these different sources of variance, we can't trust these findings. So, ultimately, what I think is a better way of thinking about this is what I call the Drake rule. So instead of using race as any kind of a marker of group differences, it might be easier instead, let me see if I can make this smaller. It might be easier instead to use specific exposures and mechanisms as a marker of group differences instead. So measure the thing that you're measuring rather than trying to find some sort of approximation. Moving quickly onto some discussion of Lasagna's law, which states that the incidence of patient availability sharply decreases when a clinical trial begins and returns to its original level as soon as the trial is completed. Another way is saying that we as study PIs have no idea where our patients come from. We have no idea where our participants come from in our studies. We just are out here doing our best, and our best is usually not very well. There's a corollary to Lasagna's law called Munch's third law, which is take however many people you assume you will be able to recruit for your research study, divide it by 10, and that is a much more realistic estimate. Now, although these tongue-in-cheek laws were developed with respect to overall patient recruitment, they apply doubly or even triply when it comes to diversified and underrepresented populations. So the question that we're trying to answer is, why can't we recruit diverse populations? Now, this is my own picture of an event that took place here in Boston a few years ago, pre-COVID, that was marketed to Black individuals. It was in the Black community. We had it in Roxbury. And in previous years, this event had drawn over 300 people. And so they planned for 300 people, but as you can see, there were about 7 that actually attended. So the question is, can we understand why something like this happened? Is there a way to sort of quantify that effort or even intervene on it so that we don't end up embarrassing? Somebody actually lost their job over this event over at the Alzheimer's Association. So ultimately, the question is simple. Why can't we recruit diverse populations into our research studies? It doesn't really matter the area that you study. It doesn't matter whether it's neurology or cancer. It doesn't matter whether you're talking about recruiting individuals to your study team or recruiting populations into your study itself. It tends to be some combination of these 11 factors. The challenge, though, is knowing which of the 11 factors are operating most concretely or most significantly with your study, and whether you have resources, expertise, or tools to address it. So when it comes to thinking about this work as a science, the field is currently deeply fragmented. We don't necessarily have clear definitions or boundaries or any sort of comparative effectiveness in this space. There are lots of confounds, like what exactly is a minority even? If you think about the COVID-19 data, so recruited by Pfizer and Moderna, they reported racial and ethnic minorities from a global trial in which white people are actually the minority. White people are about 16% of the population. But even though the trial was recruiting on 6 continents at the time, Blacks and Latinx individuals were still reported as minorities, even though that's only true in the United States. And then ultimately, and I think most significantly for those of us who are doing clinical research, there's no real infrastructure in order to do this. So if you are thinking about trying to improve the diversity that you have in your studies, and you recognize that the field that's responsible for getting this right is fragmented, there's lots of confounds, there's no sort of infrastructure, you might sort of have this reaction. You might say, you know what, peace, I'm out of here. I'm just gonna leave this alone and do what I've been kind of continue to do. However, I am here to help. I'm here at Mass General with you, and it turns out that there are some solutions. The first thing that I want to recommend is taking those 11 reasons, those big 11 reasons that seem to be operating in most of our clinical trials, and modeling into an ordered workflow, something that we can recognize. So it turns out that instead of just saying, I've got $500 in my budget, I can maybe address one of these. Think about this from a way where we can map this onto some sort of ordered workflow that helps us understand where the selection bottlenecks are potentially coming from. So what I want to propose here is that instead of thinking that your research study starts at this level of screening and enrollment and then moves on to a setting, implementation, and retention, that there are many decisional stages that individuals need to navigate, or they need to be either sufficiently privileged or literate to navigate before they are coming into our research studies. So what this means is that the areas in green here are the ones that we typically have robust protocols for in our research studies. Everything else is typically left up to chance or somehow separated or segmented out from the formalized order of our research studies. So we're leaving a lot of information up to chance, which is why recruitment tends to be difficult for everybody, and recruitment of special populations tends to be especially so. So now what I'm going to do is talk a little bit about some data from my team at the Care Research Center that highlights ways that we have found to measure some of these elements of variance, and then ultimately how we have managed to overcome them. So let's start with selection bias. Now, in 2018, I was approached by the Michael J. Fox Foundation, who said, we have run this online study because we figured it's too hard to get people into subspecialty clinics. We want to improve the diversity of our study. And so we have run about 20,000 people who may have a Parkinson's disease through this online digital study called Fox Insight. And here are the demographics. And so in case you can't necessarily read the label at the bottom, it's white for a reason. And if you can't understand the graph, just got Bernie Sanders here to help you interpret the quality of the data. We know that Parkinson's disease may be more likely in white individuals, but it's not this much more likely. And so again, the problem is here that, it's not a matter of direct healthcare access because it was online. All of you can go online right now and sign up for Fox Insight. So why do we have these kinds of demographics? And it turns out that it wasn't just a matter of race. If you looked at the median annual income, the median participant earned about $105,000 a year, which is well above the national median at the time of about $58,000. And 1 in 3 individuals had a doctoral degree, which again is not terribly representative unless you think Parkinson's tends to mostly affect white men who have a high income and are wildly overeducated. So, that's just not realistic. So clearly, there's some kind of selection bias here. So working with the Assistant Director of the Care Research Center, Angie Sanchez, we designed something called the Fire Up PD study, where we took this ordered workflow and we focused on these early measures, thinking very carefully about sampling selection, who we're gonna be inviting into that study, making sure that we're raising awareness, building trust, and building literacy in order to drive recruitment into this online study. So the study was really designed to focus on multiple barriers. We had a really broad definition of minority. So as you might imagine from those charts that I just showed you, Parkinson's research studies are really, really good at recruiting rich, white, overeducated men who live on the east or west coast of the United States. So we basically said everyone else is a minority for purposes of this study. We had a really strong focus on the sampling frame and the protocol. So we had individual sites sort of identify a key demographic group that they wanted to reach, a barrier that group particularly experienced, and an intervention that would resolve that barrier for that population. And we randomized 8 sites to intervention or control within pairs. We gave them about $45,000 to intervention sites, $10,000 to control sites for a 5-month intervention. And here, the goal was not necessarily to improve recruitment. The goal was just to try to understand, can we get individuals to that stage of making a decision, showing some kind of interest in the study? And so the interventions that were involved in the study were mostly focused on community and physicians. So either engaging with that minoritized group that they were trying to reach, or reaching out to minority serving physicians in order to raise the profile of the online study. So again, even though this study was not designed with any kind of primary outcome of recruitment, nevertheless, here's the baseline of where we found in terms of accrual to digital study for those 8 sites. And what you see here is control sites, even though they were passive control conditions, they weren't supposed to be doing anything other than what they were already doing. During the period of the study, they nevertheless saw a huge spike in non-white enrollment to their studies. And intervention sites saw a huge spike, not just in non-white enrollment, but also in individuals who were low income or low education. And what's notable about these latter 2 columns here is that these individuals were white. So even though they were not targeted, we saw that for intervention sites, there was nevertheless kind of an across the board improvement in accrual to this digital study. We also, so even though we were able to recruit about 500 individuals, we also saw that for 230 individuals, so about 50%, were not able to participate. And so usually when we talk about ethno-racial minorities, we talk about the factor of mistrust. And as you can see here, the stated reasons for not being able to participate were less about mistrust and more about time commitments, the digital divide, and a lack of language literacy. So unfortunately, for this particular study, you had to speak English. in order to participate. Unfortunately, lots of individuals who have Parkinson's don't speak English, and so therefore they were rendered ineligible. We also recognized that there were problems with the digital divide. So recognizing that the problems for participation aren't necessarily where we think they are was a really key finding for this study. And so we've actually been able to follow it up to really understand that mechanism of action. Again, we did not predict any sort of significant difference in enrollment for the study, but nevertheless, we still found it. Why did we find it? Why did we also find it in control conditions, even if they were ostensibly doing the same thing? So what we have done is we have in the sequel study, which is going on currently, we've harmonized those community engagement activities across intervention and control sites. So everybody is doing community engagement to see if that is the mechanism of action, or whether the mechanism of action may be restricted to the intervention condition, which will still be doing that bespoke intervention that's designed to reach a certain population. And so you can see that we basically are doing a pre-post design where we're doing the harmonized activities, surveying individuals, and then we are moving to the intervention or control condition, and then surveying people again to see if there's any sort of marginal difference for intervention sites over control sites in that latter recruitment. So the study kicked off just this past March. We finished up the harmonized community engagement in July. We are currently in that control and intervention period. So fingers crossed that by the end of the year, we do have some emergent data around the study. Moving forward into thinking about measurement error as an important source of variance here. We also did a project recruiting for the ADNI-3, which is this large sort of multi-site study that is looking at trying to get a detailed neurological biomarkers and cognitive markers for individuals that may be eligible for Alzheimer's disease clinical trials down the road. So across 4 sites, Emory, Kansas, Rochester, and UCLA, all across the United States, we basically did sort of a detailed analysis of these 5 dimensions that may indicate their ability or likelihood to recruit, in this case, Black and African Americans, to the ADNI-3 study. And so what we ended up finding was that there were certain sites like Emory, which placed a high value on community engagement and partnerships down in Atlanta. They didn't necessarily max out the grid across all of these, but they were really, really good at that. There were sites like Kansas, which had really great workflow and metrics. They made it really, really easy to identify a population that was ready for sampling. And then those individuals were very carefully and clearly funneled into appropriate research opportunities. There were sites like Rochester, which if any of you are working with the University of Rochester on one of many neurological studies, you know that they tend to be a recruitment powerhouse. But on these particular dimensions, while they do have a tightly integrated workflow and metrics, they don't necessarily put a lot of time or effort into some of these other dimensions that tend to be important for recruitment overall, but especially for diversified recruitment. And then you've got other sites that kind of sort of maximize other kinds of ideals. And so what you can do is you can kind of take that data and use it to build a mini sort of inverted pyramid that's similar to what I was highlighting earlier. So a site like Rochester, with their tightly integrated workflow and metrics, weren't necessarily great at being able to reach a highly minoritized population. And then the ones that they did actually encounter, they were not great about being able to hold on and recruit from that group. And so then by contrast, you see that a site like Emory, which does tend to do well when recruiting racial and ethnic minorities, was able to reach a large number of individuals and then retain them through each of these individual stages for a relatively high percentage of those, or a relatively high fraction of those individuals who were ultimately enrolled and recruited into the study. So when you're comparing sites that are really good at reaching some of these populations, it's not just that they have one person who's really good at their job or they're doing one thing particularly well. They're basically having multiple checkpoints along multiple decisional stages that end up making it work. And so what this means is that you can take this framework that I've highlighted before, and you can basically use it to identify key bottlenecks for your studies. So instead of saying, I can't get anybody to participate in my study, you can ultimately understand, is it a problem of sampling frame? Is it a problem of raising awareness, building trust or motivation, or a case for equity or return of value? rather than just saying that we're struggling with overall recruitment. We're really breaking down these early sort of pre-screening phases to help you understand where the bottlenecks and potentially facilitators for your studies are coming from. Very quickly, we also did a really great project that was funded by PCORI, focused on modifying a theory of change. So instead of approaching community with a very specific recruitment protocol or study in mind, we approached Black Bostonians. This study was led by Helen Hemley, who's up there in the corner, to try to understand how these communities would like to be approached by research. And so she convened 12 stakeholders. They met every month for a year and gave us a lot of really key insights about recruiting for clinical trials in Boston. And I hope that you all appreciate that Boston is a really, really unique environment. It's a relatively small place with a lot of academic institutions conducting a lot of clinical trials. So even though Black individuals don't necessarily make up a large part of the studies in the MGB system, these communities are approached really, really frequently for clinical research. And so what we found out here is that there is a certain weariness among Black Bostonians because they remain the same, but the faces that approach them continue to change, and they don't seem to have learned from the last people that kind of came calling. And so what we ended up doing was we've developed really, really robust tiers of engagement. So instead of thinking, that we are engaged with a particular community, we have a much more robust ordered workflow that helps us understand what kind of relationship you have built within certain community members, within certain community institutions and community-based organizations. And so what we found here is that there's a necessity to invest and really build these relationships over time and to ensure that there is a return of value that speaks to the people that you're trying to recruit. And it turns out this kind of thing, not just what researchers conceive as having a return of value, as having value, is important for overall recruitment. So even though we found this for Black Bostonians, and of course it would apply to this particular community, if you are designing a research study, of course it makes sense that the people that you want to take part in your study perceive some sort of value. We need to think about value as being broader than contributions to science. And so this particular group developed a really, really great thing called an outcome map, which we're working on publishing now, which basically takes kind of your standard logic model and really supercharges it by thinking very carefully about stakeholders, transparency, dissemination, and evaluation. So we can actually think more broadly about this. If you think about work from social science, we know that all of these things that I'm discussing, whether we're talking about sources of variance or other kind of factors here, may intersect with aspects of individual, team, and sort of systemic levels. And so a colleague of mine, Andrea Gilmore-Bykowski, talked about this model of building partnerships that may someday lead to research participation. And so what I want you to kind of potentially recognize is that, given the model that I've already presented to you earlier this morning, the Gilmore-Bykowski sort of dimension is just a more detailed look at these stages of building trust, building literacy, equity, and return of value, by talking about prioritizing different foundational aspects of building and sustaining those relationships over time. My work with the NI Impact Collaboratory, which is focused on pragmatic trials in dementia, you often uses something called the PRECISE-2 as a kind of a way to assess whether your trial is kind of a classic RCT design or whether it's more pragmatic in nature. So there are 9 domains in the PRECISE-2, and it's usually used as a backbone to determine whether there's strong real-world evidence for your particular intervention. One of the things that's really key about pragmatic trials is that it tends to randomize healthcare systems rather than individuals. And so many people say, well, for randomizing entire healthcare systems, that we don't have a diversity problem. We don't have an equity or an inclusion problem. Unfortunately, that's not necessarily true. So we need to recognize that, while you can have traditional outcome measures across multiple components of any sort of large framework that's conducting clinical research, there are also equity outcomes that you can integrate into each of these groups as well. So instead of thinking about equity as a standalone, one-time offer of maybe going into a Black church and talking about the necessity of trusting researchers, we can recognize that there are lots and lots of ways to quantify these efforts across different dimensions of design, analysis, and maintenance of our research studies. And so just as it is true in pragmatic trials, it's also true in canonical classical research studies. Very quickly, I wanted to talk about some of the work that we have done with the EPPIC-Net clinical trial network, which is focused on pain management, which is headed up here at Mass General. So if you take the same framework, it turns out that there are specific activities that you can use to emphasize some of these different levels. So depending on where your bottleneck is for recruitment or retention into your study, you might consider one or more of these options to help resolve them. So if you have a trouble in terms of screening enrollment, so maybe your eligibility criteria is disproportionately balancing out people of color, a brand identity campaign, which is designed to optimize awareness, is not going to work for you. Instead, you might want to think about building patient focus groups to help you understand and identify and resolve any sort of perceived burden or challenges in recruitment that you're having at that level of pre-screening or screening. So you need to make sure that the intervention matches the problem and resolves it at the right time. In the same way that we tend to design classic interventions for clinical research. If you're trying to intervene on the problem of diversity, equity, inclusion, you need to have a solution that matches the problem that you're trying to relieve. It is just good science. So ultimately, what I want to kind of end on is a little bit of a kind of a glimmer into the work that I'm doing as part of the Pioneer Award that I received about 2 years ago. And it really is predicated on a simple question. How can we measure and understand the difference between an event like this versus another event that my community that I ran, again, pre-COVID, where we ended up having standing room only. So the room that we reserved for this event was full, and unfortunately we had to turn away about 60 individuals from participating. So, that's not necessarily a good thing. It just means that we rented the wrong size space. But how do we understand the difference between that first event from this event? They were held, for the same community within a mile of each other here in Boston. What distinguished these events from one another? And so that is why I launched the Forever AD study. Forever is a really long acronym that means the Foundations of Reliable Engagement, Valid and Effective Recruitment, which is really focused on operationalizing and quantifying recruitment, engagement, and retention in dementia studies. So it's really focused on trying to underpin elements of selection, measurement, and implementation. So it's divided into 3 phases, which are itself, each one is roughly the size of its own, or kind of an R01, where we're assessing lay communities. So we're trying to understand different sorts of barriers, knowledge, and attitudes about Alzheimer's disease itself and dementia, and then also what people's attitudes are about research. Instead of going to individuals who have engaged in research, individuals that are adjacent to large academic healthcare facilities, we're using basically political canvassing. We're basically ripping off Barack Obama's 2008 kind of community canvassing model and doing it in the name of science. And so the goal is to do this in Harris County, where our goal here is to do a relatively large survey of minoritized individuals of about 15 to 16,000 individuals. In the Alzheimer's space, this is about 20 times larger than any available study that's looking at these same questions. We are also using that data to develop advanced mapping tools using the floating catchment area metrics to understand where we can identify not just barriers, but also facilitators to reaching certain minoritized populations. Phase 2 of the study actually kicks off in a couple of weeks with a trip to St. Louis, where we're going to be doing basically an ethnological assessment of 20 Alzheimer's disease centers of excellence around the United States. We're going to be developing bespoke outreach dashboards and workflows for sites to use. So basically, in other words, we're going to use the data from phase 1, the data that we gather locally, to build a dashboard that helps sites identify where those bottlenecks are, where they're performing well, where there are emergent opportunities, and where ultimately their recruits are coming from for research studies. We're going to be piloting that at BU, but if any of you want to use this for your studies, I'm happy to write a diversity supplement or an administrative supplement to extend to whatever your area of research is. Phase 3 is designed to put these 2 things together. So we've got the lay community on one side, the research community on the other. We're going to bring them together and develop a kind of a community partnership curriculum that we will be workshopping here in Boston if you would like to take part. And we will be working with an outreach platform that was put together by NIAA called Adore to help contextualize the data that we find there. So in conclusion, what does this all mean? We need to recognize that when it comes to thinking about recruitment and engagement, these things are not black boxes. You don't have to guess. You can be scientific, you can be thoughtful, you can metricize your efforts. While that work is still ongoing, we have made a lot of headway within my research center, and recognizing that this is really just the tip of the iceberg. We're going to have to start to bring in data from epidemiology, library information sciences, bioethics, social theory, and may, I think, represent a novel field of research, which I'm currently calling representation science. So the idea that you can pull from all these other areas to build a science that's focused on these relatively well-identified populations. Now, we often tend to equate diversity, equity, inclusion, but I wanted to sort of clarify for purposes of what you're probably trying to do with your research studies. Diversity is a stopgap. It is not the goal. It is a stopgap on the, for this broader concept of inclusion on the way to what I think we can all agree is probably the appropriate way to design our studies, which is to be as representative as possible, as generalizable as possible. And so we need to make sure that, that we have representative samples in all that we do. There is a strong scientific justification to do this. Obviously, there's a moral imperative, but if you don't have a representative sample, it does call into question the validity of your findings. Unfortunately, that's just kind of the way that it is. So in my opinion, if we start to pay attention to these things, carefully build out the science, then we will soon see that equity will be a primary driver of innovation. We're already starting to see that in the private sector. There are lots and lots of startups focused on building diverse registries, AI models that are leveraging big data. As you might imagine, having deeply representative and generalizable samples is a key function on the way to building personalized and precision medicine. So I do think that within the next 5 to 10 years, these are problems that we are all going to have to start to be thinking about. I'm happy that that my research center has the opportunity to kind of lead the way or build the path. But my hope is that everybody will be taking on some component of this in the next decade. So, if you forget everything that I've said here today, all the data that I've showed you, all of the components, I really just want you to remember the Drake rule, which is to use specific exposures and mechanisms as your marker differences, rather than relying on something as old and tired as self-indicated race. So many, many thanks to the many people that make up the CARE Research Center. Any of the individuals with an asterisk represents what we call our community trust. These are individuals that actually are responsible for the governance of the center. They're not rich, they're not scientists, but they are individuals with deep community ties to various parts of Boston. And of course, my grant support there. So that is the end of my formal remarks, and I'm now open for any questions that people might have. Thank you. Yes.
So, great job. Few things I'm still starting to reconcile, but I guess one would be, you look at all those pre-enrollment steps that you outlined, trust engagement and all that. Mm-hmm. At the end of that, to what extent do you end up with a representative sample? And to what extent do you end up with the rich doctors of the Michael Fox Foundation? Are you selected for people who want?
Yeah, so the question for those of you who are online is really focused on, if you believe that inverted triangle, if you follow all the steps and do the best that you can, does that necessarily guarantee that you'll have a representative sample, or whether you'll have a sample full of rich white doctors who are taking part in the Parkinson studies, for example? And so that is a really, really great question. And what I want to do is maybe I can back up to a version of this and kind of walk you through it. Everything in your question depends on a well-defined sampling frame. And as my research center does free consultations, by the way, so if you're ever struggling with recruitment, please come see us. We're part of the Division of Clinical Research. Everything depends on a well-articulated sampling frame. So if your sampling frame is poorly defined, saying that you just want to reach people with knee osteoarthritis, for example, then what you're going to tend to find is individuals who are sufficiently literate or privileged to kind of skip all of these steps, screening enrollment, are the ones that are going to approach you for the study. If you are more careful about defining your sampling frame, and you've ensured that you have a design that is flexible, that allows people who may not normally be able to participate to kind of get in the door. So sometimes that means, enrolling on the weekends or staying past 5 o'clock at night, as like one minor example. Then you might get more of that representative population into your study. What's really great about this framework is that it's not just a bottleneck for overall recruitment, but it is a bottleneck that can help you identify where that representation starts to be constrained. So let's say that you have a really well-defined sampling frame that helps you understand, all right, I want to reach this, I want this population to be fully generalizable. But then by the time you get to the stage of literacy and motivation, you realize that everybody's really, really privileged. Then you've lost some aspect of equity in those intermediate stages. And what I encourage you to do is to create some sort of intervention within your engagement and recruitment process that allows you to reach those individuals a little bit more specifically. And so instead of just wondering at the level of pre-screening or screening, with this slightly more nuanced model, you'll be able to identify very, very concretely where that privilege is starting to creep in and be a determinant of participation rather than the eligibility criteria that you've identified. Yes?
Well, first of all, thank you for the presentation, for the work. It's unbelievable the amount of ideas and action that you've been deploying with your groups and colleagues. So I have two somewhat simple questions, but first is what's going on with education for children, health literacy, that helps them understand when the importance of this participation in trials and how this is recruitment. And the second question is whether community health centers could be funded to be clinical trial sites, and what's the impediment or the challenge that you would foresee?
Yeah, so those are two, I think, spectacular questions. So the first one, for those of you who are online, was talking about building literacy, especially literacy around research in kids. And then the second one was leveraging community health centers as effectively as change agents to help promote kind of diversifies research studies. The second question is actually easier to answer than the first, which is using community health centers. If you think about how community health centers could be leveraged, community health centers would be really, really great at helping you build awareness for a particular research design. They tend to have high levels of inbuilt trust and engagement with their treated population. And they can also work with you as key study partners to build that literacy and motivation and a strong return of value, because there is kind of this local loyalty that is built into places like community health centers. So what I would recommend is that we should be embracing community health centers as sites for clinical research studies. There are some challenges there. So number one is that clinical CHCs tend to be busy. The doctors there tend to be overworked. So you would need to either make some changes to the infrastructure of the way that community health centers are currently funded and structured so that they have the time, the space, and sort of the incentives to do research. And that's where you start to think about things like equity and return of value, not for participants, but for other organizations who participate in this kind of work. So this model is very generic because it can be applied to multiple stakeholder groups and sectors. So if you can properly incentivize community health centers, you're moving in the direction that I think we're going to continue to move in, which is decentralized clinical research studies, where maybe you come into Mass General to do something that needs an on-site cyclotron, but for everything else, for the cognitive assessments, for a blood draw, you can go to your local Walgreens, you can go to Whittier Street Health Center, for example. And therefore, you can sort of sidestep a lot of these challenges that may spell, that may make it really difficult for minoritized populations to come into, a place like Mass General to participate. The first question is about building literacy for clinical trials in kids. And this is a little bit tricky because clinical research in pediatric populations has a fundamentally different set of challenges than clinical research for adult populations. So if you are trying to improve literacy for kids to participate in clinical research at that age group, there doesn't tend to be that same level of trouble that we often have. So pediatric oncology clinical trials have by far the highest rate of consented participation of any kind of clinical trial design. And I'm sure you can imagine why. But then there's also sort of the factor of kind of building in literacy building for the parents or the caregivers of those children. So what might be better is leveraging, again, a decentralized approach and some sort of brand identity campaign to build awareness in a general population, to build literacy in 4 sectors that I think are really important. So building research literacy, building health literacy, but also building literacy in 2 key areas that I think are often overlooked. One is medicine as distinct from health, and then the 4th one is insurance. Because if there is a study going on at Mass General Hospital, but your insurance means that you won't normally get your care at BMC, then you're not going to look twice, even if insurance isn't a factor in participation. So building literacy on these 4 key areas, health, medicine, research, and insurance, I think is a really, really key phrase. I think that would be better pitched at an adult population. But obviously it's never too early to kind of start building this even in younger populations. See if there's any questions in the chat or on the Q&A. I'm not seeing anything, but please feel free to type something if you're on the Zoom. All right, any other questions or thoughts? All right, thank you very much. All right, thank you, everyone. Carol, do you want me to wind things down? Or?
Thank you so much.
Thanks so much.